Let us look at the question. So the figure 4.1 or 4.0 shows the vertical section of the vascular system in the dicotyledonous plant or stem. So this is also a dicot, and we are told it's a stem. So now, and again, the key thing is that the vascular system is made up of the xylem vessels, and of course phloem vessels. Now, where we are coming from, we raised or we answered a question, and it's very important to go back and look at the question, this question, which letter represent the structure in which substance move either up or down. So in the phloem, substance can move up or down. But since we are talking of the vascular system here, we know that one is a phloem, the other one is a xylem. And now let us see the question. Identify the structure T. Now, how are we going to identify the structure T? And I must admit that this question is also not clear, but I'm going to, by all means, explain so that you understand the thing. So now, identify structure T. So here in between there's a cambium, that separates the two. You know that. So there's a cambium here in between that separates the two. But there's one here and the other one here. Now, in this S, you see, the arrows are pointing up, which means the substance are just going in one direction, up here. And here they are going down. In the xylem, you never find things moving downward. And therefore, we can identify T as the what? The phloem. Okay? Identify structure T. So this is the phloem. Now, aside from the substance moving downward, right here at the end of it, there are cells here that you are not able to see. There are cells here. And these are what you call companion cells. Okay? So now, what two structures or features in the diagram that helped you to identify the structure or to identify T as a phloem? So we've identified as phloem. What made us to identify it as a phloem? So one, movement of, so one is the arrow. Let me put arrow. So the arrow showing the direction of movement of substance. So one arrow showing the direction of movement of substances. Okay, the direction of the arrows, where are they going? Downward. So that is one feature that helped us. Then aside from that, presence of what? Presence of companion what? Cells. Only xylem, only phloem only phloem have companion cells. Okay? Companion cells. And of course, in the way we are coming from, I'm sure we are going to find again the other question that is asking us about concerning companion cells, as we are going to see. Name two substances transported in S. What are the two substances transported in S? So S, we said, is the what? The xylem. Two substances transported in xylem, we said xylem transport water and what? Mineral salts. Very important. So water and what? Mineral salts. Now, what about if it was in structure labeled T? What are the two substances? So one, sugar or glucose. Then also amino acid. So we might have answered the question that you might encounter as we go on. So take note of these answers. And of course the cambium, you know the function, where we are coming from. Okay, I hope you've not forgotten. We said it carries out cell division and also separates the xylem and the phloem. That is question. Let us go to question C1. On the diagram, label the companion cells. So I've just done here. So the companion cells just needed to put an arrow here, not really circling them, but just to put an arrow. That is what you needed to do. So this one ends as a mark. Explain the role of the companion cell in the vascular bundle. What is the role of the companion cell. Okay? So what is the role of the companion cell? So listen very carefully here. So companion cell provide energy for active transport of substance. So provides energy for active transport of substances. What are the substances that are transported actively? Amino acids and glucose. Because these, they are transported up or down. Okay, so they need, or the cell need a lot of energy, or needs energy. So that is the role of the companion cell in what? In the vascular bundles. That is very important. And of course, from cell specialization, if you had time to look at my video on cell specialization, I explained much on the companion cell. So if you have not done that, please get to look at the video on cell specialization where we answered questions. Then now, explain two functions of the cambium chi. This one, we've already answered it. And of course, I'm just from explaining here. So carry out cell division. Okay, carry out cell division to produce new cells. What are the new cells that are produced? Xylem and also what? Phloem. Very important. Then now, what is the other one? The other one we said is separates xylem and what? Phloem. Okay, or separates xylem from the phloem. So that is the function. Okay, or these are the two functions. Okay, let us move on to look at another question now. Let us move on to look at another question. So this question came in 2021, if I'm not mistaken, 2021 GCE. Okay. Then now, the figure 3 shows parts of the organ of a plant. So now, the first thing that you need to know is to identify which organ of the plant is this one coming from. So this is the internal structure of a leaf. So the organ is a leaf, in case we find the question. Then now, what are the questions? A, that is Roman numeral 1, identify the structure G and K. So what are the structure G and K? So now it's very important here to look at it very carefully, because there's another structure here that is a thin layer that is on top there. And this one I would label, it is called the cuticle. Okay? And it has a function. Okay? One of the function is to prevent, or to prevent, that is excessive loss of water from the leaf. Okay, and it is shiny. One thing to know. Okay, what about G? This next part here, it is called the upper epidermis. So G is the upper epidermis. Upper epidermis. Which means there's also a lower epidermis. So this is called upper epidermis. Okay. Then what about K? So K, where is K? So K is right here, and there's an opening here between. So the cells between here, the ones that cause, or the one that forms this opening, these are called guard cells. So that one is called guard cell. Now this opening, just to name it, is called the stomata. Okay, this is stomata. Then state one structure difference between structure G and D. So structure G, which is the epidermis, and J. So J is the whole of this one. So this one is the lower epidermis. So what is the difference between the upper epidermis and the lower epidermis? How many? So one. So one difference is that, look at this. This one, the lower part has what? The guard cells. Okay? It has the guard cells. That is one thing that you should do, understand. So structure J has stomata, while structure G has no stomata. So structure J has stomata, while structure G does not have stomata, or does not have. Let us look at now, identify the part organ, or the plant organ in figure 3.3. So we're answering question B. identify the plant organ in figure 3. We've already identified it. This is the leaf. So the plant organ is the what? The leaf. In which labeled region, that is labeled region 3, or in which labeled region in figure 3 is the rate of photosynthesis the highest? Where is the rate of photosynthesis the highest? So rate of photosynthesis is the highest in region H, and this is where you find, this is the palisade, this is the palisade mesophyll. So this is where you find the palisade cells.